Okay, so the very, very simple thing that we have to try to figure out is how a single cell in your body knows where it is. If you think about it, each of the cells in your entire organism is producing a different set of proteins. Yet they do this with the exact same DNA and with many of the very same environmental signals. So, eyes here. So, if I have a signal, if I have a cell in my pinky, it is likely experiencing many of the same things that a cell right next to it is also experiencing. However, they act differently. Think about how close your eye is to the skin, how close your nail is, your hair. Think about the fact that you have stripes and holes in your face. Right? Okay? Those all have to happen somehow. And it's really difficult to explain that with traditional gene expression. Now we know that there are other things. We know that there are internal environments in cells. And we know that DNA replication can carry over epigenetics as well. So we know that there's more to it than that. But what you don't realize is how finely tuned gene expression is. We think of things in terms of gradients, or we think of like a stinky man standing in the corner of the room producing some odor. And we think that if you are here versus here, you are experiencing relatively the same environment. I'm smelling about the same, right? Here, stinky. Yeah, pretty much equally stinky. But in a cell, that difference in response can be huge. It can create an entirely different set of proteins. So how do we accomplish that? Well, this is gene expression, like the real way. Now, I don't remember where I got this from, but it looks very British. Is anybody confused by anything we're saying here? No. Just tell me if you're confused.
Isn't it okay?
Go back. We have DNA. We're getting copied into RNA. We have transcription. This is all different ways of looking at the same thing. We've got RNA polymerase. So these, you can actually see here, is DNA along the center here. And these are all different pieces of RNA that are being copied off of it. Kind of cool, huh? I don't feel like we need to go over translation again, splicing. One idea is important probably that we kind of forget. RNA is highly unstable, both from a structural point of view and from the point of view that the body destroys it quickly. So RNA does not stick around. So after RNA is produced, it's quickly destroyed, which is why we don't see the same kind of effects in gene expression as we do in, well, why we don't see lasting effects in gene expression. Okay. Now let's break it down to the structure of a gene. Genes have basic parts, and it's important to think of those parts as functional units. There's the promoter, which is the region designated right before this arrow. So this is a piece of DNA. This is DNA right here. Same thing. This and this. Same exact thing. This purple thing is this purple thing. This gray thing is this gray thing. This is a promoter. Does it look any different than this sequence of DNA? No, they look identical. What is different between the promoter and a regular piece of DNA is the sequence. There will be parts of this DNA that transcription factors will specifically bind to. The question that I was just asking you, how does a single cell in your body sense it? How does a cell that is right here know that it's supposed to be an eye cell? It's the same question as this. This is what you look like when you are first born. You are essentially a tube. This is your brain. Different parts of that tube will expand, giving you the structures that you see as the brain. The blue here, yellow, green, so forth. So this becomes this becomes this. How do you get differences in gene expression, drastic differences in gene expression between your brain and your spinal cord when they're so close to each other. We'll come back to the slide. Okay. It's a little confusing. Let's break this down. Let's see what we got here. These are cells. That's the inside of the cell. This is the outside of the cell. Assume that something is being produced over here, a signal, and there's a lot of it over here and very little here. Lots of signal over here, little signal over here. This cell can sense that signal, which means it's got a receptor on the surface of it. So let's look at what happens. When the signal binds to the receptor, a transcription factor is activated. This is a transcription factor. What we have here is 3 different promoters on actually the same piece of DNA, but we've broken it apart just so you can see them. Now, this promoter has a single binding site for a transcription factor. This promoter has 2 binding sites for a transcription factor, and this one has 3. That means, in order for this gene to be activated, or in order for this promoter to have 3 transcription factors bind to it, there's going to have to be a higher transcription factor concentration. What? Okay. All right. One more time from the beginning. Signal from the outside. Transcription factor is activated. If I have very, very low amounts of transcription factor, I'll fill this first slot in all 3 promoters. If I have more transcription factor activated, well, then I'll be able to fill the second slot. And if I have even more, then I'll have 3 slots filled. What does that mean? These 3 promoters respond to the exact same transcription factor, but they respond to different concentrations. This one requires a very low level of transcription factor to be activated. It'll almost always be activated. This one requires 3 times that. So, what we just said over again. Here we have a signal. We have a low amount of signal. A single transcription factor, single, it's not going to be ever single, it's going to be thousands or millions. Transcription factor was activated. That's enough to bind to just one of these things. That means protein A is going to be expressed in this cell. With a higher level of signal, more transcription factors are activated. Protein A and B are expressed in this cell. Higher transcription factor, higher signal, lots of transcription factors, all 3 genes are activated in this cell. That means one signal, one type of signal, let's call it sonic, the sonic signal, was able to create 3 different patterns of gene expression. Same epigenetics, same genetics, same signal, but different amounts. So now we know, we add this to the story that we have, we know that with the same environment and different epigenetics, we can produce different proteins. We know that with the same genetics and same epigenetics, same external environment, we can have different internal environments that will produce different proteins. And now we see, with the same epigenetics, with the same genetics, with the same internal environment originally, the same signal, but in different amounts, we can produce 3 different patterns of proteins. So why is this interesting? Well, this is why it's interesting. Because the story that I just told you is not one that's made up. It's completely real. me simplify this diagram for you. Let's say we have a signal. Oh, I don't know. Let's call it Sonic Hedgehog. Now, let's say it's produced right here. So, let's just say we produce amount of 5 there, 4 there, 3 there, 2 there, 1 there, and 0 there. Now, let's pretend that at level 3, eyes are produced. Everybody with me? The whole thing is a cross-section of your head. So if I were to take his head, slice it like this. Okay? So this is a cow and not a human. If you were a human, you know, the eyes might be up here. The signal is being produced here in the front of the head from a certain group of cells. Okay? So it's actually being produced along this side. Right? Okay. Well, what if I reduce the amount of signal being produced? So what if I were able to make it so that instead of a level 5 being produced here, we have 4. 3, 2, 1. Where would the eyes then be? Here. And what if I were able somehow to produce a poison that reduced the amount of Sonic Hedgehog being produced so that 3 was right there? What would that look like?
Cyclops Cow.
Cyclops Cow.
Cyclops Cow. Hellebore is the poison which shuts down Sonic Hedgehog production. Sonic Hedgehog is produced right here. Because so little is produced, that level 3 is here, which means the eyes form on top of each other and together. You can look it up. There's Cyclops Cow, Cyclops Cat. There's a few Cyclops examples. Anything that will consume this hellebore.
It's like one body.
The important thing to take from this is not Cyclops Cow. This is how you're going to remember it. This is the tag that I'm putting in your head that you will never forget. The lesson that you are supposed to get from this is this. These promoters each have a region that's similar. In order to activate the gene they're attached to, I can use different amounts of transcription factor. So a low concentration can produce different signaling amounts. Now, we could have B inhibit A, we could have C inhibit A. It doesn't matter. The important thing, the thing that I really want you to understand is that you have sensing mechanisms, concentration gradient sensing mechanisms in your DNA. And what those can do is sense a particular level of signal. Let's say A and B is what produces the I. Right? So, it's a super fine-tuned balance of signals. It's not like you would have more I if you have more sine. No, there's no I. There's no I at levels 2, 1, or 0. There's no I at 4 and 5. It's a super finely tuned balance. You need exactly that concentration to produce the I. Right? So this takes gene expression to another level. When I ask you to produce your organism, when you're coding it, when you're trying to figure out how to make a cell that is right here, different from a cell that is right here, you're going to need a concentration-dependent promoter. Almost all of you are going to use it. I can almost guarantee it. This is the mechanism. All right? I think I have another example. It's not nearly as exciting. Let's cut it there.